For the fifth part of the adaptive immune response lecture, so we will talk now on the antigens. So antigen is like the identification card for the pathogens. So this is how our immune cells recognize the foreign or the invaders. Antigen is the combination words of antibody and generator. So now it is called antigen. Antigens are molecular structures that are able to activate the immune response. The chemical structures can be in the form of carbohydrates, lipids, nucleic acids, proteins, or combinations of these molecules. Organisms that cause a disease, the pathogens, possess a variety of parts that may contain antigen, such as can be observed in the Salmonella bacteria. They have the pili, cytoplasm, ribosomes, flagella, plasmid, DNA, cell wall, the capsules, and many others. And for viruses, for example, in the coronavirus structure, we have the S protein, the spike envelope, the RNA, the N protein, nucleocapsid, the membrane protein, all is can be recognized by our immune cells. And each type of pathogens has its unique antigen. So different types of viruses have different or their own unique spike protein. So even though they look the same, but each type of viruses have their own unique structure for every parts of their antigens. As for the coronavirus, now it is around close to 200 potential candidate for vaccines in development. So all the vaccines is produced in order to stimulate our immune system to produce response against the different types of antigens in the COVID-19 virus. So we hope in the near future we can the scientists will be able to produce one that effective enough to be used for us. So let's have a look on the one of the most common disease, demam or the common cold. So why don't we ever develop immunity against the common cold? So we can, in our lifetime, even once a year or even every month, we can suffer from this common cold. Actually, we do develop the immunity, but the issue is there are more than 200 viruses. This we talk about viruses. Bacteria is another story. So if we talk on viruses, there are more than 200 types of viruses that can cause the common cold or demam. This includes human rhinoviruses. There are around 100 types of rhinoviruses that can cause common cold to us. Coronaviruses, parainfluenza viruses that cause the influenza flu disease, and also adenoviruses. So if we focus on the coronaviruses, it is for the coronaviruses, there are 7 types of coronaviruses known to infect humans. So even in the coronaviruses, we have 7 types. From them, 7 different types, even the name are the same, but they have totally different antigens on them. So the patients infected by these viruses will develop respiratory symptoms of various severity, means either it is like a common cold, normal flu, demam biasa, or can be very severe, hospitalized in the ICU or even death. So 4 types of coronaviruses are associated with the common cold, and the other 3 coronaviruses, such as the one that cause COVID-19, severe acute respiratory syndrome, the SARS-CoV-2, or the SARS-CoV, the one that caused SARS epidemic in 2003, and the MERS that caused the Middle East respiratory syndrome somewhere in 2000, epidemic somewhere in 2012. So now 2019 is the time for SARS-CoV-2. These 3 types can lead to more severe respiratory tract infection, as we can observe in the cases of COVID-19, which can potentially be lethal. All coronaviruses share very similar structures, but each with their unique antigens. So this is how we differentiate them into 7 types, because all of them have 7 different unique antigens on their structures. This transmission electron microscope of the COVID-19 virus. So now we have a look on the antigens. So antigens actually consist of many epitopes. It's the small structures on the antigen. So we can observe here one molecule of antigen have different epitopes. So this is the part where the antibodies especially will bind or identify the antigen. Epitopes are small part of a large antigen. Antibodies and T cells recognize epitopes. So antibodies and the T cell, they do not recognize the whole antigen, but they will recognize the epitopes on the antigens. So the Fab or the antigen binding site on the antibody will specifically recognize the epitope, and also the T cell receptor on the T cell antibody, they will bind to the specific epitopes. They will not bind to the whole antigen, but to the specific epitopes. So a single antigen, one antigen may possess many different epitopes. Different antibodies may bind to a different epitopes on the same antigen. So as we can observe here, one antigen have 3 different types of epitopes, different types of epitopes. So the body can produce 3 different types of antibodies just to bind to the each epitopes on the same antigen. Example, protein as the best type of antigen molecules contain many epitopes on the surface, because their protein is a very large molecule. And for structure, the bacterial flagellum is a large complex protein structures. So they can possess hundreds or even thousands of epitopes on them. Immunogenicity is the ability of an antigen to provoke an immune response. So different antigen got different levels of ability. So this means that not antigen have the same ability to induce the immune response. Some is very weak, and some antigen they are very strong. Once only just introduce one, so they can provoke a strong reaction. But some antigen introduced to the body will not provoke any immune response. So in general, the more complex the molecules or the bigger the molecules, the higher the ability of that antigen to provoke an immune response. For example, antigen with strong immunogenicity, such as proteins, carbohydrate, we can observe here the top parts is the one that have better immunogenicity compared to the lower part. So proteins is the best. Second is carbohydrates, lipids, nucleic acid, heptanes, and amino acids are very poor antigen because they have a very low immunogenicity. So the ability for nucleic acids, heptanes, and amino acids to provoke immune response is very low. So antigen with strong immunogenicity, they will be able to stimulate both the humoral immunity and the cell-mediated immunity, such as antigen made of large protein, or just able to stimulate production of antibodies, such as carbohydrate. Antigen that able to induce immune response is called immunogen. So this means all immunogens are antigens or antigenic, but not all antigens are immunogenic because some they are very weak, they are not able or poor ability to provoke the immune response. So why the word why here we in a sentence called all immunogens are antigenic, of course, but not all antigens are immunogenic. So this cause we can observe in the case of heptane. So heptane is the molecules that are too small to be antigenic if they are alone by themselves. Heptanes are essentially a free epitopes that are not part of the complex 3D structure of a large antigen. So for example, these are the heptane molecules. They are free epitopes. They are not immunogenic enough, so the heptane in free forms cannot stimulate the immune response. For a heptane to become immunogenic or able to stimulate immune response, first it must stick to a conjugate or pair itself to a larger carrier molecule to produce a conjugated antigen. So if the heptanes stick to the carrier molecule, so now they become immunogen. So once the heptane combine with the carrier molecule, so they become bigger, so the immune response now can identify them. So now they have the immunogenic ability, and then once they have a good immunogenicity, so they will be able to produce or stimulate immune response. The heptane specific antibodies produced in response to the conjugated antigen are then able to interact with the unconjugated free heptane molecules. So once antibody is already being produced to the heptane, so the next time, even without the carrier molecules, because our body already produce antibodies to it, the antibody still can detect the free heptane because it is already being stimulated against it. Example, many cases in allergic responses. In penicillin reacts chemically with proteins in the body to form a conjugated antigen. The penicillin conjugated antigen can cause allergic reactions to drugs in the penicillin class. This common penicillin is a very common, commonly found in the prescribed antibiotics. When you went to the clinics, there is higher chance that the doctor will prescribe penicillin to you. So these are the cases where people develop allergic reaction to penicillin class of antibiotics. So that's why it is mandatory for the doctor to ask whether you do you allergic to any drugs or not? So this is one of the reason why the doctor asks you that. Second, people that are allergic to milk, so similar mechanisms with lactose, where lactose combined with the proteins, carrier proteins, to become conjugated antigen, so the body produce antibody to the lactose. So the second time the person drink a milk, so the body already have antibody against the lactose on that milk. So this will lead to immune response. So allergic response actually the immune response. So we have two type of antigens in the adaptive immune response. The major histocompatibility complex, the MHC antigens. So proteins are found on the surfaces of cells that help the immune system recognize foreign substances. MHC antigens can be found in all vertebrates. Their main function is in the cell-to-cell interactions. For example, between the MHC class 1 with antigen to the T cell receptor. So those are one type of cell-to-cell interactions. MHC in humans also called human leukocyte antigen. So this MHC, the same thing, if they are in human, also known as HLA. MHC class 1 is expressed on surface of almost every cells in the body, whereas the MHC class 2 is restricted to surface of the immune cells in the phagocytes especially. Every person come with their own unique MHC. So everybody, almost every person in the world possess unique MHC, just like your IC number. So it is unique to you. So it is very rare to find people that have the same IC number. You can say, some say it is impossible. It is very rare for two individuals to have the same set of MHC molecules, except for twins, the identical twins, because they come from the same cells. So let's look at the MHC class 1. MHC class 1 especially allows a natural killer cell and T cytotoxic cells to recognize foreign cells attached on the abnormal cells, MHC1. MHC class 1 presents endogenous antigens, for example, viral proteins, proteins from intracellular bacteria, fungi, protozoa or other parasites, or tumor or abnormal antigens. Healthy cells will present self-antigen from its own cell by displaying it on the MHC. So if the cell is healthy, they still will display the antigen, but they will display the normal antigen to be recognized by the T cells. Infected cells presents antigen from a pathogen, or tumor cancer cell will present abnormal or altered self-antigen, or they will not even show any antigen on the MHC. So this is how the NK cells and T cytotoxic cell will become alert or recognize there is something wrong with the cells. Either it will display the pathogen and the antigen from the pathogen, from the virus, bacteria, etc., or it will display abnormal antigen on it, or they will not even display anything. So when that three things happen, it will detected by the cells, the immune cells, where we bind to it and will start the killing process. If the cells are displaying the normal antigen from the self-antigen, the normal self-antigen, the T cell bind, recognize them as the normal cells, so they will be spared. They will not be killed. So the processing and presentation of endogenous antigens. First, for MSc class 1, they focus on endogenous antigens, which is produced within the cell itself. For example, here we can say in the case of virus infected cell, the virus will go to the nucleus to replicate. Once the nucleus produce a viral, when the nucleus ordered the cell to produce more viral proteins, the cytoplasm will degrade the protein into peptides or digested antigen. So this broken down antigen into peptides will be moved into the endoplasmic reticulum, where here in the endoplasmic reticulum they will bind to the MSc class 1 molecule. To produce peptide class 1 MSc complex, so which then move through the Golgi complex to the cell surface. For MSc class 2, this allows T cells to recognize antigen fragments attached on antigen presenting cells which possess the MSc class 2. APCs present exogenous antigens such as coming from bacteria, fungi, protozoa, free viruses, etc. The antigen presenting cells will display the antigen fragments of the pathogen by sticking it to the MSc class 2 on its plasma membrane surface. So this is how they display the process antigen. Processing and presentation of exogenous antigen in MSc class 2. So first, the exogenous antigen is engulfed by the phagocytes through phagocytosis or endocytosis. Phagocytosis is one of the endocytosis process. Second, it will enter the endocytic processing pathway, which the antigen is digested or degraded into small peptides by the enzyme lysosome. So the lysosome will fuse into together with the engulfed antigen. So this is the part where they will be digested. And then the process or the digested peptides will be presented onto the class 2 MSc molecules on the cell surface by the antigen presenting cells. So here they will engulf, process, attach to the MSc class 2 and then displayed to be recognized by the T cells.